Okay, so we're going to move right along here on the Lecture 7 series. This is, again, the second half of Chapter 7. We've already given a brief introduction of some of this material in the previous chunk. Two more chunks to go in the Lecture 7 series. So what we've already talked about is phosphorylation and zymogens as potent forms of regulation. Phosphorylation, of course, is reversible regulation, and zymogens being an irreversible regulatory mechanism. And as promised, where we're going to move in this chunk is to active sites. We're actually going to talk about an enzymatic mechanism. An enzymatic mechanism is a very detailed description of the chemistry going on inside the active site, where we can understand the stepwise methodical process of the catalysis. So we're going to be looking at a fairly simple enzymatic mechanism, one of the simplest enzymatic mechanisms known, but as you'll quickly realize, even a simple enzymatic mechanism is not all that simple. We'll be using chymotrypsin as our example, the same digestive enzyme that we used as an example of a zymogen. And then in the last chunk of this lecture series, we'll talk about the different types of catalyzed reactions in general, setting us up for some of the different types of reactions that we'll see in the second half of the course as we move on towards metabolism. We'll end with a brief discussion of coenzymes, these small biomolecules that help enzymes do their job. So if we're going to be talking about an enzymatic mechanism, we're going to be talking about the active site in detail. We're going to be getting a very clear and detailed view of the chemistry that goes on in the active site. To remind you, the active site is the part of the enzyme, the cleft of the enzyme where substrate binds and where the catalysis actually happens. It's here in the active site where the substrate is pummeled and pounded and stretched and torn and ripped into this physical thing, this physical entity, which we refer to as the transition state, the halfway between state where substrate is really no longer reactant, but it's not quite yet product. Remember, the transition state is the bread dough between flour, water, eggs, and salt, and the actually fresh baked bread. Once transition state is formed, it's highly energetically unstable, it's a very high energy molecule, and it instantly collapses into product. It goes down that free energy diagram, releasing all that pent up energy, and so product is formed. Since the active site is not evolved to bind to product, the moment product is formed, it's released by the enzyme. The enzyme itself is unchanged, it goes back to being an empty enzyme, and it can float off and bind a new substrate molecule and repeat the process again. So it's fair to say that all of the chemistry and catalysis of biochemical reactions that are catalyzed by enzymes occurs in this relatively small cleft or region called the active site. And the chemistry and catalysis of the active site, because it's part of the enzyme, the protein, depends solely, and not surprisingly, on the three-dimensional shape of that active site. It all comes down to structure equals function, to three-dimensional shapes dictating what occurs. In this case, what we really mean is that the chemistry and catalysis occurring in the active site depends on the exact three-dimensional shape and orientation of the side chains of the amino acids that make up the active site. And so this leads us to wonder about a few different things. First, if the side chains and their orientation in the active site makes all the difference, what are the amino acids that are present in the active site? What are those amino acid side chains? What are the three-dimensional orientations of those side chains with respect to one another? And what is the mechanism of the reaction? In other words, what are the steps of the reaction? What happens first? What happens next? How do we get to transition state to eventually form product? So enzymatic mechanisms are best described as methodical step-by-step processes of reactions. What's going on in the active site once substrate is bound? What happens first? What happens next? What happens third? When do we make transition state? How and when does that transition state collapse into product? So we'll do this with chymotrypsin. We're already somewhat familiar with chymotrypsin. We discussed it in the last lecture chunk. It's a digestive enzyme. It's a protease. It's responsible for cleaving peptide bonds, and it digests proteins in the small intestine. Please remember that we make it as a zymogen. It's synthesized through the process of transcription and translation as a zymogen, and we call that inactive form chymotrypsinogen. Once it gets into the small intestine, chymotrypsinogen is cleaved by pepsin, That makes it pi-chymotrypsin, and pi-chymotrypsin then cleaves other pi-chymotrypsins, fully activating them into chymotrypsin. So we know a lot about chymotrypsin. We know how it's made, we know where it goes, we know how it's activated, we know it's a zymogen, and we also know quite a bit about its enzymatic mechanism. We understand, as biochemists, how chymotrypsin cleaves peptide bonds. It's a fairly simple process. It's just 6 steps, and so we'll use this as our example of an enzymatic mechanism. Although we'll be going into some fairly detailed discussion of chymotrypsin, and we'll talk specifically about things that chymotrypsin does, please keep in mind that the principles in general that we're discussing here, in other words, this step-by-step way of looking at enzymes, how side chains play into the chemistry, what goes on to substrate when it's in the active site, all of those generalities are applicable to all enzymes that we find in our bodies. The details, of course, can be quite different, but the general ideas and premises are the same. First generality is that all enzymes require a reactive group in order to interact with and bind to, in other words, capture substrate. And this reactive group it's hungry, it's antsy, it wants something, it's unsatisfied. A reactive group, because of this unsatisfied nature, because it's hungry for something, hungry for charge, hungry for electrons, hungry for hydrophobicity, because it wants something, it's reactive. In other words, it will do something if you let it. Reactive groups typically are charged groups. There's no more reactive group than a charged group. A positively reactive group is very hungry and antsy to interact with something negative. But they aren't always charged. We can have hydrophobicity as a reactive property as well. Alright, so we know that we have an active site. We know that chemistry is going to occur in that active site. We know we have to bind to substrate. And we know that we need something reactive in that active site that is going to force chemistry to happen. So let's back up just a moment and consider the generalities of amino acids. Amino acids in general, when they're free, are reactive molecules. They have charge. The amino group is positively charged and the carboxy group is negatively charged. But that's only when amino acids are free. When amino acids are present in peptide chains, they're not reactive in and of themselves. The positive and negative charges of the amino and carboxy groups are neutralized through the formation of the peptide bond. So if we want some kind of reactive component to an amino acid, we have to find it in the side chain. Once an amino acid is part of the peptide chain, it loses its native reactivity that we would find in the amino and carboxy group. So we need to go for reactive side chains, reactive variable groups, if we need reactive amino acids in the active site. So these are our 20 amino acids, and some of these are more reactive than others. I'm sure a number of you can already identify or pick out the more reactive amino acids based on nothing more than what we've already said. So those of you who are looking in the direction of aspartic acid, glutamic acid, lysine, arginine, and histidine, good for you. Those are our most reactive amino acids because charge groups are the most reactive, and those are our charged side chains, the acidic and the basic amino acids respectively. In addition to amino acids with charged side chains, serine, cysteine, and tyrosine are also quite reactive. We won't go into the details here, but you'll notice that all 3 of those have hydroxy groups. Hydroxy groups in the side chains are polar. They have strong partial charges, and we can phosphorylate these, as we've talked about in the previous chunk. And once you add a phosphate group to these hydroxyl groups, you make them negatively charged. So there's partial charges inherent in these 3 amino acids, and the potential to make them fully negatively charged through phosphorylation. I should point out that every single protein starts off with an amino terminal end and ends with a carboxy terminal end. So every single protein, every single polypeptide chain is positively charged at its very, very beginning and negatively charged at its very, very end. And there's some potential for reactivity there as well. Alright, so remember the purpose of an active site is to initially bind to substrate and then to work on that substrate and become transition state. And we need reactive side chains, reactive amino acids in that active site to allow that to be possible. In that process, by providing the energy working on the substrate, the active site and the enzyme as a whole contribute some of the energy necessary to reach the transition state. So the way that enzymes lower delta G double dagger, the way that enzymes lower the energy of activation is by the enzymes themselves providing the energy and doing the work of reaching transition state. Once that occurs and transition state is reached, you don't need any more energy inputted into the system. The rest of the reaction is energy releasing, and so that happens spontaneously all on its own. So you can think of this as we start with some amount of innate energy contained in the covalent bonds of the reactants, and we have to put even more energy into that system, and that is the energy of the enzyme pulling and ripping and twisting and torquing and distorting the substrate. And once that substrate reaches transition state, that transition state automatically, spontaneously becomes product. Either it cracks, or it changes into something new, or it makes a bond with another substrate molecule, but all of that is energy releasing, and so we have an overall negative delta G. Transition state spontaneously collapses into product. So let's talk now specifically about chymotrypsin. As we said a little while ago, chymotrypsin is a protease. It eats proteins or digests proteins in the small intestines, and as part of this overall digestive process of the foods we eat, of course the proteins we eat have to be broken down as well, broken down into their independent amino acids. So that's what chymotrypsin is aiding. Chymotrypsin does nothing more than hydrolyze peptide bonds. It's its only job. It takes some water, and it uses water to break or cleave the peptide bonds that hold individual amino acids together into the proteins we've just eaten. You can have chymotrypsin do that same job and cleave peptide bonds on some artificial substrate that you've created in a laboratory, and you can monitor chymotrypsin when it's in the middle of its work and figure out what it's doing. And in fact, that's what has been done in order to make the material that I'm about to share with you known. So scientists studied chymotrypsin in a lab, in a tube, and kind of fooled it into digesting these man-made or people-made artificial substrates so that we can watch it in real time. Enzymatic function in general and the overall monitoring and determining of enzymatic mechanisms is an entire subfield of biochemistry. Biochemists everywhere are trying to determine enzymatic mechanisms for enzymes that are clinically and physiologically relevant. So for chymotrypsin, amino acid number 195, that's the 195th amino acid in the chain of chymotrypsin, happens to be a serine. And that serine is absolutely required for the activity of this enzyme. Remember, serine is a reactive side chain. It's not fully charged, but it has one of those hydroxyl groups. And we know that serine 195 is required for chymotrypsin activity because if we go into the DNA sequence of the gene for chymotrypsin and we mutate that serine codon into another amino acid, chymotrypsin can no longer function. It can't cleave peptide bonds. So that serine is critically important for enzymatic activity. What's more, this observation puts chymotrypsin into a pretty exclusive group of enzymes. Chymotrypsin is a member of what is called the serine protease protein family. These are proteases that cleave peptide bonds through the action of a serine amino acid. So chymotrypsin is a member of that group. Trypsin is another member of that group as well. A second very important amino acid in chymotrypsin is amino acid number 57, the 57th amino acid in the chain, and that amino acid happens to be a histidine, a charged amino acid. Histidine we've spoken about pretty much since the beginning of this course. It has a positive charge at near physiological pH. These two residues, serine 195 and histidine 57, and pretty much only those two residues out of the total 245 amino acids that make up chymotrypsin are pretty much all that's important for the activity of the enzyme. So if you were a biochemist and you saw that these two amino acids were so important, would you guess that they were in the active site? You probably would. It seems as though these two amino acids are critical for the catalysis of chymotrypsin, and indeed, that's so. Using X-ray crystallography, the three-dimensional shape or structure of chymotrypsin has been determined, and indeed, we see serine 195 and histidine 57 smack right there in the middle of the active site. Take a moment to appreciate the incredibleness of what we're looking at here. We're looking at a rendering of the true three-dimensional shape of chymotrypsin. This is what chymotrypsin looks like in real life. Most of the peptide backbone has been indicated by this green ribbon. The side chains have been removed because it would clutter the diagram too much. Here we see in the center of the enzyme is the active site. And look who's side by side, right there in the active site. Serine 195 and histidine 57. Just as our mutational analysis predicted, these two amino acids are critical for catalysis because they are the big players in the active site. Aspartic acid 102 seems to be important as well because we see that aspartic acid 102 is right there in the active site as well, and aspartic acid is a charged amino acid. It's an acidic amino acid, so it's reactive. Histidine is charged, aspartic acid is charged, and serine is reactive, as we just told you, because of that hydroxyl group in its side chain. These are all reactive amino acids with reactive side chains that are very likely to be participating in chemistry, and there they are in the active site. This general premise that we're covering here is actually another important point for enzymes in general. For the most part, as crazy as it sounds, entire enzymes, and chymotrypsin is no exception, entire enzymes exist solely to position a very small number of amino acid side chains in a very specific and exact orientation in the active site. Kind of, period. That is to say, enzymes are largely scaffolding needed to put very specific and few side chains in very specific places right there in the active site. These three residues, histidine, serine, and aspartic acid, and only these residues, in this particular orientation, and only in this orientation, is what allows this enzyme to achieve the catalysis that it needs to achieve. Said simply, in general, enzymes exist exclusively for their active sites. The entire enzyme exists to create the active site it contains. The rest of the enzyme, really, is nothing more than glorified scaffolding to achieve that active site. I like to think of this analogy in this example. What we see here is a robotic surgery machine, or a surgical robot. The robot, the surgical aspect of this robot, is right here in these three prongs. You can program this, or a surgeon can control this robot to perform very, very precise, small-scale surgeries on a patient. This robot is huge. It probably weighs close to half a ton, I would guess, and it's very, very large. But it's doing very, very small-scale, fine surgical procedures that all occur in this very small space. I think you would agree with me when I say that the rest of this large robot exists exclusively to place and control these very small probes right here in this region. In other words, all you need to perform the surgery are these small probes. But you can't just make a robot containing those probes, because those probes have to be very precisely controlled and placed in three-dimensional space, and that's what the rest of this robot is built for. Enzymes are exactly the same. All you really need for the catalysis are the very few side chains of the amino acids in the active site. You can think of this as the active site. But the rest of the enzyme is needed to place those side chains in that specific orientation in three-dimensional space. So chymotrypsin as an enzyme can pretty much cleave any peptide bond, but it prefers peptide bonds that lie immediately after large nonpolar amino acid side chains. And so therefore, very close to serine 195 in the active site is what we refer to as a hydrophobic pocket. Essentially, there are hydrophobic side chains close to and exposed to the watery outside environment, close to and exposed to the watery environment near serine 195. What happens is that these hydrophobic side chains in the active site are very unhappy. They're exposed to water and they hate that. Large nonpolar side chains in substrate molecules are also unhappy if they are exposed. These large nonpolar side chains in the substrate molecule will seek refuge and, quote unquote, air quotes, and will interact with the hydrophobic pocket of the chymotrypsin active site. Essentially, the large, bulky, hydrophobic side chains of the substrate molecule will shield and shelter themselves from water by huddling up with the hydrophobic pocket of the active site of chymotrypsin. That brings the peptide bond adjacent to those hydrophobic residues right in line with serine 195. This properly orients the peptide bond in the active site so that serine 195 can get to work. This brings up another general principle about enzymes. Much of the properties of the active site are more about lining up substrate molecules, atoms, and bonds properly so that the chemistry can be as easy as possible. Substrate binds in only one possible configuration so that everything about that substrate is situated correctly, and then the enzyme can get to work. The analogy I think of here is a drilling machine. When you're making precise holes in substrate, in your metal or your wood, you spend a lot of time placing that substrate, that board, into the machine. You measure and adjust and place and measure again and adjust and get everything just right. You get everything situated in three-dimensional space exactly correctly so that then, once everything is lined up, you can do your thing and drill that precise hole through your substrate. Enzymes work the exact same way. Substrate is bound in the active site very carefully and precisely so that everything is lined up just right, and only then, zip, will the catalysis occur, and it will occur properly. So we'll end this lecture series by moving on to the chymotrypsin mechanism itself. I think you will agree that we've kind of covered quite a bit here, though, in leading up to that. To summarize what we've talked about, we gave some general principles of biochemistry and enzymatic reactions by saying that the chemistry and catalysis that occurs in the active site really is depending on the precise configuration and orientation in three-dimensional space of the side chains of the amino acids in that active site. All enzymes require some reactivity in that active site, and usually that comes from the reactive side chains of charged or polar amino acids. Enzymes have a limited number of things they can do, a limited number of tricks up their sleeve, and we'll see a good number of those tricks when we talk about the chymotrypsin enzymatic mechanism itself, and we'll fill in the remaining gaps of what are some other things that enzymes can do biochemically as we wrap up that last chunk. But until then, as always, thanks for watching.